Hello dear students. So again coming to the competency 10.2, describe and discuss the functions and properties of synapse and reflex receptors. In my last presentation we've started reflexes, spinal reflexes. We talked of important component of reflex arc. We talked of muscle spindle and the stretch reflex. So continuing with the previous presentation, I will again repeat the specific learning objectives that the end of the session, not exactly this session, but all the three presentations on reflexes, you should be able to define, reflex and outline the components of reflex arc clearly, which we have already studied in the last presentation. Then differentiate between monosynaptic and polysynaptic reflexes appropriately. We have studied the monosynaptic reflexes. We'll be studying the polysynaptic reflexes here, and the difference we'll be studying in the next presentation. Then enumerate different spinal reflexes. We have already done that. Physiological basis of stretch reflex and its detail we have already gone through. Different spinal reflexes with their physiological basis. We'll be covering most of the spinal reflexes today only in this presentation. Then role and structure of muscle spindle and Golgi tendon organ. Muscle spindle we have discussed in the last presentation. Golgi tendon organ will be studying in this presentation. Alpha gamma motor linkage, neuronal linkage we have already studied, and the role of higher centers in control of spinal reflexes will be studying along with this presentation, then in subsequent presentations. And in this we'll be also be studying the properties of spinal reflexes, abnormalities, which some of the abnormalities which we have already studied in the previous presentations we'll be studying subsequently in other presentations. So now I'll be talking about the second type of sensory receptors of the muscle, or the proprioceptors of the muscle. This is the Golgi tendon organ. Golgi tendon organ, or the neurotendinous organ, or the other name is tendon spindle. These give information to the spinal cord about the change in the muscle tension because of their specific location. The Golgi tendon organ, or what we call as GTO, is an encapsulated sensory receptor through which the muscle tendon fibers pass, and it is located immediately beyond their point of fusion with the muscle, that is at the musculotendinous part of the muscle. It consists of group of nerve fiber endings which are non-myelinated, which is present in the form of a beaded or a knobby appearance in collagen tissue covered with a capsule. Talking about the dimensions of Golgi tendon organ, each Golgi tendon organ is about 1 millimeter long and approximately 0.1 millimeter in diameter. Now there are major differences between muscle spindle and Golgi tendon organ. One of the differences is its location. Muscle spindle is present in the belly of the muscle, while the Golgi tendon organ, we know it is present in the musculotendinous portion of the muscle. Another difference is that a single Golgi tendon organ is connected in series with 10 to 15 muscle fibers, while the muscle spindle is connected in parallel and it is present in the belly of a muscle. Secondly, another difference is that there are less number of Golgi tendon organ in a muscle as compared to the muscle spindles. One more difference is that there are no muscle fibers in the Golgi tendon organ, while there are intrafusal muscle fibers in the muscle spindle, and the muscle spindle has the efferent nerve control of gamma motor neurons, while the Golgi tendon organ doesn't have any efferent control. But the similarity is that they have a sensory innervation. Now these are being innervated, or the impulses from Golgi tendon organ are mediated by group 1B nerve fibers, which are rapidly conducting, large diameter about 16 micron, and they are myelinated nerve fibers, and they relay in the spinal cord, they go to the posterior root or the dorsal root of the spinal cord, and then they enter the spinal cord, and they have dual ending. One of the ending is that they end in the interneurons in the spinal cord. These interneurons are stimulated by the Golgi tendon organs, and then these interneurons they relay on the alpha motor neurons where they secrete glycine, and glycine is an inhibitory neurotransmitter. So these Golgi tendon organs, they are going to stimulate interneurons, and interneurons are going to inhibit the alpha motor neurons. The second termination is in the neurons in the dorsal horn of the spinal cord, where they second order neurons, they go as spinocerebellar tracts and other ascending tracts to the brainstem reticular formation and cerebral cortex, and through this spinocerebellar tracts, the central nervous system, it comes to know about the location of the moving part of the body, that is the muscle. Golgi tendon organs, they form the backbone of a very important reflex, Golgi tendon reflex. This is a protective reflex. So whenever a muscle is stretched, it responds with contraction. More stretching, more harder the muscle will contract. That is, the muscle becomes more hard and more contraction. But then there is a limit. Then there is a physiological limit beyond which the muscle cannot further contract, because if it contracts beyond that point, what will happen? The muscle is going to get avulsed from its origin or insertion. And so body has been developed such that immediately when the contraction goes beyond that physiological limit, the contraction stops and muscle relaxes. So this response is called as the Golgi tendon reflex. So here, because it is opposite to stretch reflex, it is also known as inverse stretch reflex, and it is controlled at the local level of the muscle, that is the sensory receptor present in the muscle itself locally controlled. So it is called as autogenic inhibition, that is the muscle controlling itself, and thus muscle, because when the muscle relaxes, it lengthens slightly, so it is also known as lengthening reaction. Now let us see that how the Golgi tendon organ sends impulses. So this is the Golgi tendon organ present in the musculotendinous part of the muscle. This is quadriceps femoris muscle. So whenever the quadriceps femoris muscle is stretched, it causes contraction and extension of the knee. When there is too much tension on the quadriceps muscle, this tension will be mediated by the or taken up by the Golgi tendon organ, and when it is beyond the physiological limit, immediately it will send its impulses via the group 1B fiber through the posterior root of the spinal cord into the spinal cord. Here it relays at two interneurons. One interneuron is inhibitory and other is stimulatory. So the inhibitory neuron, that is the neuron which secretes glycine or another inhibitory neurotransmitter at its termination, it will end on the anterior motor neuron which supplies the quadriceps femoris muscle, and immediately it will be inhibited and then the muscle will relax, that is, instead of being extended, the knee will be relaxing. But the impulses also go to the another interneuron which is the stimulatory interneuron. This interneuron ends on the anterior motor neuron which is supplying the antagonistic muscle. So antagonistic muscle here is the hamstrings or the flexors. So the knee, instead of being extended, now relaxes. So this way the Golgi tendon organ mediates the Golgi tendon reflex. Now the Golgi tendon organ is stimulated both by passive stretch of the muscle and also by active contraction of the muscle. But for the passive stretch to activate the muscle, it should be a very extreme stretch, because most of the stretch is being taken by the elastic fibers. Secondly, the Golgi tendon organ, like the muscle spindle, show both the dynamic response as well as the static response. So when there is extreme tension developed in the muscle, and the Golgi tendon organ immediately send the volley of impulses to the spinal cord, this is the dynamic response. But when After some time, the tension is being maintained or the slow sustained tension is being maintained. The Golgi tendon organ start sending impulses at the slow rate, and that is the static response. So, like muscle spindle, Golgi tendon organ shows both dynamic as well as static response. Thus, now we are clear that the Golgi tendon organ inform the nervous system instantaneously about the degree of tension on each small segment of muscles. So, the functions of Golgi tendon organ based on this Golgi tendon reflex is mainly is protective function. Any strong and potentially damaging force will reflexly inhibit the muscle. So this will prevent the development of any damaging or too much damaging tension. Otherwise, what will happen? The muscle will try to maintain the force and risk damage. But here what the muscle, instead of contracting, it lengthens and the protection occurs. So this Golgi tendon organ, it plays a protective phenomena through its reflex mediation. In certain patients who have upper motor neuron lesion due to any causes, the muscles become hypertonic. In these hypertonic muscles, there is a particular sequence of resistance to stretch is encountered, that when these muscles are moderately stretched, there is muscle contraction, and when the stretch increases or it becomes more strong, suddenly instead of muscle being contracting, it relaxes. So this sequence is maintained, that is stretch, contraction, stretch, relaxation. So now if you try to flex the elbow of such a patient who has hypertonic or spastic limb, initially when we are trying to passively flex the elbow, we encounter resistance to stretch. When we try to further flex the muscle, try to further stretch the muscle, immediately there is activation of the inverse stretch reflex, and this leads to sudden collapse of the elbow. That is, an elbow flexes. We encounter no resistance. So again, if we continue the stretch, again same thing will be happening. So there will be a particular sequence in the muscle, sequence of resistance followed by giving way of limb on its passive movement, and this clinically is called as clasp knife reflex or clasp knife effect or clasp knife rigidity. So it typically feels like if you try to open up a knife. So initially when you try to open a folded knife or jackknife, so initially you encounter a lot of resistance, and then suddenly it opens up, this gives way to the resistance. So this is a clasp knife effect. This is not encountered in the normal persons, but encountered in the patients who have the hypertonic muscles, and hypertonic muscles are generally seen in the upper motor neuron lesions. This clasp knife reflex is an exaggerated form of Golgi tendon organ reflex. This is very important in such muscles. It acts as a protective reflex to prevent any tearing of the muscle or avulsion of the tendon from its attachment to the bone. So it protects the muscle from damage in hypertonic persons. Second important function of Golgi tendon organ reflex is equalization of the contractile force among the muscle fibers. Now in a muscle which is made up of multiple smaller muscle fibers, some muscle fibers might be exerting excess tension. So when they exert excess tension, they are inhibited by the Golgi tendon organ. And some other muscle fibers which are exerting less tension become more excited in the absence of reflex inhibition. So because of this Golgi tendon organ reflex, this leads to redistribution of simultaneously the muscle load over all the fibers in preventing the isolated muscle damage on overloading of smaller muscle fibers. So otherwise what will be happening? Some muscle fiber will be exerting, some muscle fiber will cause less contraction, another more. So the sort of balance is being maintained because of this equalization of the contractile force among the muscle fibers, and the muscle can contract as a whole in a proper manner. The muscle spindles and Golgi tendon organs, they have a major role to play in the motor control of the body by the higher motor centers. So they apprise the higher centers immediately about the changes taking place in the muscle during contraction. So whenever any signal or any motor command comes from the higher centers to the lower centers to cause the effect or cause any voluntary movement, the muscle spindles, they give the impulses back to the higher centers about the change in muscle length, and Golgi tendon organ about the changes in the muscle tension. So the impulses are carried by the rapidly conducting spinocerebellar tracts, the ventral and the dorsal spinocerebellar tracts, and by other ascending pathways to brainstem reticular formation and cerebral cortex. So now the higher centers can immediately know that if there is any difference between the command and the execution of the order. So the muscle spindles and Golgi tendon organ send the feedback or send the copy of the command, copy of the executed command to the higher centers, where the higher centers can compare between the command, that is order, and actually what is happening. If there is any error or if the change is required, that is modified in the subsequent orders. Now we will be discussing different spinal reflexes other than the stretch reflex. But before studying the other spinal reflexes, we should know what are the different properties of spinal reflexes. So we are able to understand the different polysynaptic reflexes. So these reflexes can be experimentally studied in two types of animal: the spinal animal or preparation. The spinal animal or preparation can be made with the help of spinal cord transection at the cervical region, so that the spinal cord doesn't have any control from the higher centers. In such an animal, the respiration has to be artificially maintained. Another type of animal is a decerebrate animal or preparation. Here we remove the control of the cerebral cortex or the higher center from the lower brain and the spinal cord. And in this type of preparation, the transaction is done between the level of superior and inferior collicula, that is midway between the superior and inferior colliculi. Here the respiration automatically occurs because the brainstem is intact, so we don't require any artificial respiration. There are many properties of spinal reflexes. We'll be discussing only a few which are important. Some of the properties, they overlap other properties or they mediate the other properties. So these properties are adequate stimulus, delay or total reflex time, irradiation, recruitment, after discharge or after potential, local sign, fractionization or fractionation, occlusion, summation, rebound phenomena, fatigue, reciprocal inhibition, and the last three, sensitization, facilitation, final common pathway. The first property of spinal reflexes is adequate stimulus. Like in the case of receptors, we have adequate stimulus. So same phenomena applies over here in the case of reflexes also, that each reflex has an adequate stimulus, or only a particular type of stimulus will elicit a particular type of reflex. Like the stretch of the muscle is the adequate stimulus for the stretch reflex. So we need a precise stimulus to trigger the response. If you want to flex the muscle, we need a stimulus in the form of initiation of flexion. So this is an important property of the reflex. Second property is total reflex time or delay or reaction time. The total reflex time is the infinite time which elapses between the application of adequate stimulus and the response. That is, when you stimulate a particular part, the response takes time to be elicited, or the response takes some time to be generated. So this is called as the total reflex time or the reaction time. This is the time taken by the efferent impulses to travel from the receptor to enter the spinal cord, to pass through the spinal cord where the multiple neurons are present, and then reach the interior motor neuron or the motor neuron of the particular effector organ, and then the efferent impulses to reach the effector muscle and then the effector muscle to produce the response. So is this efferent, central and efferent conduction time. So if from the total reflex time, conduction time, we subtract the efferent and efferent conduction time, that is the time taken by the reflex to move from the receptor or periphery to the spinal cord, and the time taken by the reflex to move out from the for the impulse to move out from the spinal cord to the periphery. So this is also known as peripheral delay. So the time which is left in between is the central reflex time or the central delay. So that is, it is the time taken by the impulse to pass through the different synapses in the spinal cord and reach the anterior motor or the motor neurons. So as we know that a synaptic delay is about 0.5 millisecond, that is time taken by the impulse to cross a synapse or pass through a synapse is 0.5 millisecond. So in a stretch reflex, the central reflex time is 0.5 millisecond. So we are clear that it is a monosynaptic reflex, that is only one synapse is involved. But in the polysynaptic reflexes, the central reflex time is more than 0.5 millisecond. So if we know the total reflex time and we also know the peripheral delay time, so we subtract the total peripheral delay from the total reflex time, then what time we get, we divided it by 0.5 millisecond, we get the number of the synapses involved in the reflex. Irradiation is another important property of spinal reflexes. Because of this property of spinal reflexes, we can generate a response of higher magnitude by changing the strength of the efferent impulses. So as we go on increasing the strength of efferent stimulation, the extent of reflex response increases. That is, greater the efferent stimulation, more is the irradiation in the spinal segment, that is the impulses they spread to surrounding neurons. So this will occur when there is increase in the strength of the stimulus. So the spread of impulses in the spinal cord, it spreads to more and more of anterior motor neurons as well as supra and infra segmental regions of the spinal cord. So for example, flexor withdrawal reflex, when you are pricked with a pin, you try to withdraw your, suppose you are pricked with a pin at the finger, you try to remove your finger. Suppose there is a stronger prick, that is you are pricked with a longer pin or a stronger pin, you will remove the limb totally away. So you are increasing the strength of the stimulus, you are increasing the efferent impulses, so there is a stronger response. So this flexor withdrawal reflex will occur of different magnitude depending upon the strength of efferent stimulus or the efferent impulses. Recruitment is same as in the case of recruitment of motor units. So whenever there is increase in the number of active motor units as we go on increasing the efferent stimulation. So not only the impulses they irradiate, but also there is recruitment of motor units. That is, means when an efferent stimulus is applied to cause reflex contraction of some particular muscle, and as the efferent stimulation continues, there is increase in the number of active motor units because of the recruitment and this recruitment increases the contraction of the muscle. So again repeat, increase in the number of active motor units when an excitatory nerve is stimulated with the stimulus of constant strength for a long time. So as long as we maintain the constant stimulus and increasing the strength of the stimulus, it will generate a larger response also because of recruitment of motor units, because some fibers will respond at a lower threshold and others at a higher threshold. So this increased number of alpha motor stimulation is due to reverberatory circuits, and this can be seen especially in the crossed extensor reflex, which shows a high degree of recruitment. A very important property of synaptic reflexes is after discharge or after potential. This is the reflex response of the muscle which continues beyond cessation of efferent nerve stimulation. That is, even when the stimulation is stopped, still the response of the effector muscle continues for some time. This is result of after discharge due to impulses set up in the interneurons. These interneurons continue to discharge, and motor neurons are generally stimulated via multiple interneuronal pathways, and some of these pathways take longer time to discharge. So this after discharge, which we have already read in the properties of synapse, can be of three types. There can be synaptic after discharge, that is, discharge occurs when the neurotransmitter acts for longer time, and also when EPSP remains for a longer time above the threshold level, so the neuron keeps on firing. The second type of after discharge is parallel circuit after discharge, when the neurons, they keep on discharging and then side by side stimulating the effector neuron. Third is reverberatory after circuit discharge. We can see like these in this reverberatory after circuit discharge, the impulses being transmitted from A neuron to B neuron and then to C neuron, and side by side the impulses are also being transmitted from the C neuron to the D and from D to E and F. So the circuits, they continue to discharge the original neuron or stimulate the original neuron, or we can see this also in the parallel circuit discharge, that this interneuron, let us take an example of interneuron, this is being stimulated and then it takes longer time to stimulate the effector neuron. This effector neuron B is being directly stimulated by a neuron, and also the interneurons, they are taking their time to stimulate the B neuron. So here the two interneurons are involved, so they are taking longer time to discharge. So this is the reason that even when the stimulus is removed, still the reflex response can be sometimes be seen because of this property of the synaptic reflexes. When the stimulus strength is less, the response occurs at the local level. So this is called as local sign, or the property called as local sign of the spinal reflexes. Here the basic effect is to remove the affected part from the irritating stimulus or the stimulus which is causing the response. For example, removal of limb from the irritating stimulus, like from the hot pot or from a pinprick. Fractionation is a property of spinal reflexes which tells us that, or which shows us that if we stimulate the efferent sensory nerves, the response which we get in the effector muscle is always less than the response which we get when the muscle is either directly stimulated or via its motor nerve. This is because each efferent nerve can activate only a fraction of motor neurons supplying the muscle. So this property is called as fractionation, and It occurs because of the occlusion property, because each muscle is innervated by a number of efferent nerves, and because of the property of occlusion, because of sharing of discharge zone, the impulses coming from the multiple sensory nerves, they are being shared by the sharing of discharge zone, so the resulting response is lesser. The next property of spinal reflexes is occlusion itself. Occlusion means occurs because of the sharing of the discharge zone. As the discharge zone of the neurons are being shared, the sum of reflex tension, which is developed by simultaneously stimulating maximally two efferent nerves going to a muscle, this tension will be less than the algebraic sum which we get when individually the efferent nerves are stimulated. This is because various efferent inputs are being shared by some of the motor neuron, so that there is some overlapping of the zone of activation. So we can see in this that here the A and B neurons separately, when stimulated, each is going to stimulate 4 neurons, which together comes to 8 neurons. But here the third and fourth neurons are being shared by the efferent neurons A and B. So when simultaneously we stimulate A and B, the total response we get is stimulation of the 6 neurons, not the 8 neurons, which we are going to get when we separately stimulate them. So because the two of the neurons, that is 3 and 4, are being occluded due to common discharge zone. Because of this property of summation of the spinal reflexes, the response of the effector muscles can be potentiated. So this summation occurs because of overlapping of the subliminal fringe. So stimulation of two efferent neurons together can produce greater tension in the muscle than when each is being separately stimulated. That is, response is more than the algebraic sum of the two individual responses. This occurs due to the overlap of subliminal fringe or overlap of zone of facilitation. That is, some neurons lying in the periphery are facilitated by both other neurons. Let us take an example of these two efferent neurons A and B, and their excitatory, they are converging, or sorry, they are innervating the other neurons. 1, 2, 3 are being innervated by A, and 5, 6, 7 are being innervated by B. The fourth neuron is being innervated by both A and B, and they share the subliminal fringe. That is, if we stimulate A, we will get response of only 3 neurons. We stimulate B, again response of 3 neurons, 5, 6, 7. But the fourth neuron will not be stimulated by B or separately by A, because the response, because of sharing of the by the two, so they will become in a facilitated zone. So when simultaneously both of them are being stimulated, we get a response of 1, 2, 3, 4, 5, 6, 7 neurons. Separately we were getting the response of 3 plus 3, 6 response, 6 neuronal response. So simultaneous stimulation produces a better response. So this is opposite to occlusion. Occlusion occurs because of sharing of the discharge zone, and summation occurs because of sharing or overlapping of subliminal fringe or zone of facilitation. Summation can also occur of subthreshold stimuli. So we know that we have two types of summation: temporal summation, that is the stimuli, subthreshold stimuli when separated in time, that repeatedly when we give stimuli one after other, subthreshold stimuli, they get stimulated And then we also have second type of summation is spatial summation, when simultaneously multiple subthreshold stimuli are given and they summate because of the EPSP becomes stronger and goes above threshold level and the response can be generated. Whenever a reflex activity is being elicited and then it is inhibited for some time, and again we stimulate the same reflex or try to elicit the same reflex, the response which we get after a small amount of inhibition or some period of inhibition, we get a magnified response. And this we can see when we study the flexor withdrawal reflex. We will be able to study this rebound phenomena, that is the response rebounds, it becomes more after being stopped for some time. The property of fatigue is indeed a important phenomena, because it is seen that when there is continuous stimulation it can cause fatigue of a reflex, or when there is repeated elicitation of reflex at frequent intervals, finally it will lead to progressive decrease in response or an absent response. So this property also leads to habituation, that continuously we are stimulating a part, after some time there is decrease in the response. Once get habituated to the stimuli. The site of fatigue in the spinal cord is first the synapse, followed by motor endings, and lastly by the muscle. So we need to remember that the first site of fatigue in the spinal cord is synapse. Reciprocal inhibition is a very important property of reflexes. Because of this, whenever a reflex occurs, the antagonistic muscles of the effector muscles are always relaxed. For example, when a stretch reflex excites one muscle, it simultaneously will inhibit the opposite muscles, that is here the extensors. This phenomena is called the reciprocal inhibition. Let us see in this diagram that here the impulses from the hands are going via the root 1A fiber, so the posterior route into the spinal cord. Here it is innervating an interneuron. The interneuron is further innervating the other two interneurons. Here one interneuron is stimulating the flexor muscle of the arm, and the other neuron is inhibiting the extensor muscle of the forearm. So here the effect is to cause flexion and remove the hand, and so this muscle is excited while the opposite extensor muscle of the hand, it is inhibited, and this reflex also crosses to the opposite side, and same we can see here also the opposite limb has to be extended. So here the extensors are excited and the flexors are inhibited. So this is an important property of reflexes, and which helps to give a better response. These two properties, sensitization and facilitation, they overlap with other properties, and still separately we should be able to know what are these. So when a stimulus is being repeatedly applied, the response gets intensified, and generally this is seen in the case of injurious stimuli. Here the presynaptic facilitation occurs. So when we say we are sensitized to that stimulus. So another is facilitation. Here what happens, a single motor neuron has convergence of multiple afferent neurons. So if we stimulate a single afferent neuron to that motor neuron, the response will not be that of that intensity. But when we simultaneously stimulate all those afferent neurons which converge on a single motor neuron, the response is better. So here the convergence property leads to facilitation. That is, each of them facilitates the other and produce a better response. Final common pathway. The afferent neuron or the motor neuron, they are considered the final common pathways. So because all neural influences affecting the muscular contraction, they funnel through the motor neurons to reach the muscles. The motor neurons which supply the skeletal muscles are the efferent site of many reflex arcs. So as we said that all the neural influences, they funnel through these motor neurons to affect the muscles, and that is why these are called as the final common pathway. So average motor neurons has about 10,000 synaptic knobs on them, and approximately about 5 inputs from the same spinal segment go to a spinal neuron. Also, inhibitory and excitatory inputs via interneurons from other spinal levels and from descending tracts, they reach the motor neurons. We can see in these diagrams that all the influences from the motor cortex, basal ganglion, thalamus, cerebellum, brainstem nuclei, interneurons in the spinal cord, directly or indirectly are reaching the lower motor neurons, and mainly these are the alpha motor neurons, and to some extent the gamma motor neurons are also involved, but mainly it is the alpha motor neurons which are considered the common pathway. So lower motor neurons receive signals from both direct and indirect upper motor neurons, and the sum total of all inhibitory and excitatory signal determines the final response of the lower motor neuron and the skeletal muscle. So see, this diagram clearly shows that the response reaching the muscle is via the lower motor neuron. So they form the final or common pathway. Now coming to other spinal reflexes, one of the important spinal reflexes, withdrawal reflex. It's a polysynaptic reflex, also called as pain reflex, flexor withdrawal reflex, sometimes nociceptive reflex, and this occurs in response to a noxious or any painful stimulation to the skin, subcutaneous tissue and muscles. Here the important response or the role of this reflex is to remove the body part from the painful stimulus. So all the focus is to remove the body from the painful stimulus, which can be seen in the case of a pinprick or when you touch something very hot. So sometimes if you try to stimulate the touch receptors, a weaker sort of flexor withdrawal reflex can be elicited, but that is still not this adequate stimulus for the touch receptors. The receptors for flexor withdrawal reflexes are nociceptors present in the skin, muscles, tendons or joints, and these receptors are generally the free nerve endings of A delta and C group fibers. The pathway of withdrawal reflexes, the impulses are carried by group 2, 3 and 4 group sensory group of fibers. These impulses, they first pass into the interneurons pool of the neuron, then to the motor neuron. The shortest possible circuit is a 3 or 4 neuron arc. Most of the signals of the reflex, they transverse many more neurons than this. So there are more than 3 or 4 neurons, and basically they lead to 3-4 types of circuits. Important is diverging circuits, that is the circuit, that is the impulses are spread to the necessary muscles for withdrawal, and reciprocal inhibition circuits to inhibit the antagonistic muscle, and then reverberatory circuit and control reverberatory circuits and circulatory circuit so that there is after discharge, that is the response outlasts the stimulus, and also impulses to the contralateral antagonistic muscles are there. So this after discharge will cause prolonged flexion and sometimes a series of flexion movements with a strong stimulus. A weak stimulus causes almost no after discharge. Here we can see that here the person steps on a glass and immediately the impulses, they go from his foot, then through the skin is being stimulated, impulses go to the spinal cord. While traversing through the multiple interneurons, they reach the flexor muscles to pull this foot away from this irritating focus or the stimulus. So stimulus to the flexor muscles side by side. The interneurons also stimulate the antagonistic muscles. The extensors, they are inhibited. Otherwise, what will happen? If antagonistics are stimulated, it will extend, and again you can put your foot down here. Side by side, so this is stimulation of diverging circuits to involve more and more muscles, that is flexion, and reciprocal inhibition of the antagonistic and regulatory circuits, so that even if there is no stimulus touching here, your foot should not step again on this. It remains in the flexed posture. In opposite contralateral antagonistics are stimulated. Here what will happen, that because if you lift both the foot, what will happen? You may fall down. So this other leg should support yourself. So the contralateral extensors, they are stimulated, and here the flexors, they are being inhibited. Otherwise, if you try to flex this limb, you may fall down. So you need the support of this limb. So this is extended. So the flexor will, because of this stimulation, the effector muscles, they are the skeletal muscle. Thus the response is a prepotent sort of response. So here the ipsilateral flexor muscle contraction occurs and inhibition of extensor muscles. It depends upon the strength of stimulus and location of stimulus. In a spinal or decerebrate animal, almost any type of cutaneous sensory stimulus on a limb is likely to cause the flexor withdrawal reflex, or it is a oversensitization. It's a prepotent reflex. It is a protective reflex, and so it takes precedence over any other reflex. That is, if we are stimulating or we are eliciting a stretch reflex and a withdrawal reflex, simultaneously the withdrawal reflex will be more pronounced as compared to the stretch reflex. If some part of the body besides one of the limbs is painfully stimulated, so what will happen? The response will be in the same manner, that is, the part will be withdrawn, but the reflex may not be confined to flexor muscles, even though it is basically the same type of reflex. Therefore, many patterns of the reflexes of this type occur in the different areas of the body, so they are called as the withdrawal reflexes. So thus we can say that the flexor reflex is appropriately organized to withdraw a pained or otherwise irritated part of the body away from the stimulus. So what you want? You want your body to not get damaged. So you want to remove the part from the painful stimulus, and because of the after discharge, the reflex can hold the irritated part away from the stimulus for about roughly 0.1 to 3 seconds after even the irritation is over. During this time, what will happen? The other reflexes and action of the CNS will take over and will try to remove the body away from the painful stimulus. Now, depending upon the strength of the stimulus and location of the stimulus, there are different types of responses can be seen in the flexor withdrawal reflex. So, lesser the strength, we start with the local sign. So, local sign type of the responses, local sign is that it is, we have already studied in the property of reflex that it is the ability of the reflex to confine to the portion of the body affected, that simply the body part is removed, simply taken away. Then second is flexor response. Here what happens? To remove that body part, flexor contraction and extensor inhibition occurs. That is, flexor muscles are contracted and extensors are inhibited, leading to flexion of the limb and withdrawal from the noxious stimulus. So another important part of flexor withdrawal reflex is crossed extensor reflex. It is a two-limb response. So when a strong stimulus is applied to the limb, the response includes not only the flexor withdrawal of the limb but also extension of the opposite limb and what we called as crossed extensor reflex. This is multi-segmental and polysynaptic. Here what happens is when there is strong stimulus leads to flexion and withdrawal of the stimulated limb, but it will also lead to extension of the opposite limb, and this occurs after about a gap of 0.25 to 0.5 second of the flexor response, because here the multiple interneurons are involved, so time is taken to traverse those interneurons. Time taken by the total reflex time is longer. So this is a property of part of the withdrawal reflex, pushing the entire body away from the object which is trying to cause the damage to the body, that is pushing away, that is the noxious stimuli. This occurs because impulses go to the spinal cord and they end on the anterior motor neuron to cause excitation of flexor muscles. At the same time, collaterals end on the opposite anterior motor neuron because of crossing over of impulses, and this crossed extensor reflex has very important function. It maintains the posture. Otherwise what will happen? When you are removing the limb, you may fall down. So it supports the opposite limb to maintain the posture and also moves the body away from the injurious stimuli. So this occurs if the strength of a afferent stimulus is more. The stronger the stimulus, we will get this crossed extensor reflex. Another important part we can say is shifting reaction or forelimb reflex response. This response is not a part of the flexor withdrawal reflex which normally occurs in a normal person. This can be seen in the spinal animal when we try to apply the electric shock to a hind limb of a spinal animal. So there is flexion of affected hind limb with extension of contralateral hind limb and extension of ipsilateral forelimb with flexion of contralateral forelimb. So we need to be clear that this forelimb reflex response will not be seen in a normal individual but in a spinal animal. The flexor withdrawal reflex has a widespread discharge or widespread response. When the stimulus is very strong, so we can get varied grades of withdrawal reflex because of irradiation. Irradiation, that is, we have already studied that the impulses they spill over to the multiple segments, and when the stimulus is more, why there is more and more response? Because recruitment of motor units occur and spreads the impulses, this spreads to more and more interneurons. So because of interneuronal activity, we have already studied in the properties of reflex. So the widespread result of flexor withdrawal reflex is because of the more and more irradiation and recruitment of motor units. And another important part is that the flexor withdrawal reflex has long latency, and or we can say the reaction time, because the time taken by the peripheral delay and the central delay is very long, because there are multiple neurons are involved in this flexor withdrawal reflex. So flexor withdrawal reflex shows all the properties of synaptic reflexes, spinal reflexes, like adequate stimulus, rebound, irradiation, recruitment of motor units, reciprocal inhibition after discharge. So now the next important component is that some other reflexes and the clinical reflexes with some knowledge about the upper motor neuron and lower motor neuron will be studying in the next lecture on the reflexes.